Can I start?
Yes.
Okay. Alright. Greetings to Dr. Yong and my fellow classmates. So today we will be presenting on our environmental sampling plan at Sekinchan Paddy Field. Next. Sekinchan is much notable as one of the major rice suppliers in Malaysia. In Sekinchan, it locates a vast, well-irrigated and organized paddy field. In fact, it produces one of the highest yield of rice in Malaysia. Agriculture and fishery industry has been known as their major economical activities. It also famous with its tourist attractions such as the paddy field itself, paddy gallery and Redang Beach. To maintain the production of the To maintain the production of the paddy field, of course, all kinds of methods are used and conducted which may contribute to the contamination. First, utilization of pesticide may cause many possible pollution. When its usage is not controlled, the pesticides may discharge into soil, groundwater, and nearby water bodies. Next, overuse of fertilizer also can cause problems. Firstly, it may lead to lack of nutrients in soil as it can affect the pH. Moreover, the use of some fertilizer can also affect the ammonical nitrogen and other quality of the water itself. Next slide. Rice mill can also be found in Sekinchan. This factory emits dust particle due to the activity in processing paddy harvested. The dust particle can contribute to air pollution and somehow slowly affecting the workers that have been exposed to the process every day. Lastly, open burning of rice straw after harvest season are well known in paddy cultivation. Due to this activity, it will emit smoke that may contain hazardous gases and can contribute to air contamination. There are some serious cases which involve Sekinchan paddy field. Firstly is the pesticide exposure used in paddy farming that contaminated the area, which happened on 31st January 2011. It involves the chemical called Diron that are used as algicide or herbicide, which was listed as a priority hazardous substances due to its persistency in soil and poses risk to water bodies and sediment. Next is 25 ppm paraquat was used in Sekinchan, which affect paddy physically. It is said that the dry weight, height, and root dry weight of the paddy decreased to 10, 8, and 37% respectively. So due to all of the possibilities, environmental sampling plan that includes soil, surface water, groundwater, ambient air emission, sediment sampling, sediment sampling, microbiology, fauna, and paddy sampling need to be conducted to assess possible contamination due to the factors mentioned. So enough with the introduction. I will let Adam continue with his point on soil sampling.
Okay. Hi. Next, I'll explain about the process involved in soil sampling and analysis for its physical and chemical characteristics. Next slide, please. Basically, in these two sampling, we will focus solely on the paddy field as it is the most affected by the use of pesticide and fertilizer. First of all, for sampling for physical properties, logically speaking, of course, we cannot assess the pollution by just analyzing the physical properties. However, we are still interested to analyze the physical property of soil as it can give us information on how badly can the soil be affected by pesticide and fertilizer. Some characteristics will cause the soil to be more affected. For example, sandy textured soil will have more water movement, meaning that water can bring the pollutants deeper into the soil, thus affecting the subsurface soil. By knowing the soil physical characteristic, we can have a rough idea on how badly can the soil be affected by pesticide. In this part of sampling analysis, what we are interested to find out is the soil texture, soil color, soil moisture content, bulk density, particle density, and porosity and permeability of soil. Next slide, please. Next, soil chemical analysis can be considered as the most important sampling because in this part, we can qualitatively and quantitatively assess the pollution that's happening in Sikkim Chan. For example, fertilizer are known to contain heavy metal including cadmium, arsenic, and plumbum. So in this part of sampling analysis, we want to identify the presence and the quantity of the pollutants. Other than that, we also want to analyze the overall health of the soil by determining the soil organic carbon, pH, and the total nitrogen in soil. Next slide, please. The steps that are involved in soil sampling is, first of all, we will take 15 samples from 5 sampling locations by using a hand auger. As you can see from the map, we have marked 5 locations. However, decisions still need to be made on the sampling day as we need to assess whether the marked location is suitable for sampling or not. If the paddy field is in a waterlogged condition, a new sampling location need to be chosen as it might not be suitable to take sample in a waterlogged condition. Next slide, please. The depth which sample are taken depends on the parameter of interest. Then the collected soil will be kept in a sampling bag, and if the sample is too wet, it needs to be oven dried first in the lab before storing it. And then before using the soil sample for analysis, the soil must be sieved first to remove any particle larger than 2 millimeter. The method that will be used in analysis for physical and chemical characteristic is hydrometer technique for soil texture, Munsell color chart for soil color, combustion and oxidation for organic carbon, spectrometry for heavy metal, and pH meter for soil pH. Generally speaking, soil samples are harmless. However, just to be safe, the soil sample must be disposed properly in the disposal bin as there might be presence of pollutant that will affect the local soil. Next slide, please. As a precaution when sampling for soil, proper safety precaution must be taken during sampling, including wearing suitable personal protective equipment. And then the chain of custody document must be filled and remain together with the sample from the start to the end of the process. Then do not touch the inner part of the sampling bottle to avoid introducing any contaminants. Then use only sampling bottle that has never been used. And then when storing the soil sample, do not store the sample in metal container as the material might react with the soil. Errors in sampling analysis is inevitable. For example, error that might be introduced in this process is contaminant might be introduced into the sample during sampling, handling, transporting, storage, and preparation of soil. So to avoid introducing any error, especially during sampling, the sampling method is adapted from United States Department of Agriculture, and proper handling of soil sample must be conducted. Lastly, do not seal the sampling container so that the bacteria in the soil will be in its natural aerobic state. That's all for sampling analysis for physical and chemical characteristics. Next, I will pass it to Farah to explain about the groundwater sampling. Thank you.
So, next slide. So the main objective of groundwater sampling include to observe impact of fertilizer contamination in groundwater. Next, to observe the risk of heavy metals such as arsenic that comes from excess use of fertilizer. And lastly is to analyze target element for heavy metal, total suspended solids, pH, total dissolved solid, dissolved oxygen, and ammoniacal nitrogen. Next slide. So the rationale which include the parameters that I have mentioned in the objective are as shown in this slide, where DO assess whether chemical from urea fertilizer has effect on dissolved oxygen in groundwater. TDS Determine substance from fertilizer that passes through soil and affects the groundwater. PSS, to determine portion of fine particulate matter remains suspended in groundwater. Target element. Determine exact heavy metal present. AN, determine amount of ammonia in groundwater. And lastly, pH, to see the effect of excessive fertilizer to pH. Next slide. The experiment will be conducted at tube well located in Sungai Air Tawar, Sabak Bernam. We have chosen this place due to some rationale, which includes geographical condition of paddy field is not suitable to conduct purging, where elevation hello. Okay. Okay. The experiment will be conducted at tube well located in Sungai Air Tawar, Sabak Bernam. We have chosen this place due to some rationale, which includes geographical condition of paddy field is not suitable to conduct purging, where elevation of the area is too low with level of 10 feet. Possible for seawater to mix with groundwater, which will cause error. And it is difficult to collect groundwater sample as water might be locked by clay soil due to high water retention. Next slide. So for procedure, there are two main procedures that we need to follow. Firstly, collecting samples. In this procedure, there will be two equipment that will be used separately according to the type of sample. First is baler. Baler is used for sample that will be analyzed right after sampling, which are pH and DO. So for the procedure, collect sample using baler. Using baler by fully submerging it into the tube well. Then remove the baler carefully from the water and quickly bring to the surface. Samples need to be transferred into the sample container, and last, the sample are then analyzed using the YSI 556 MPS. Okay, second, next slide. Next slide is by using peristaltic pump. It is used to collect samples that need to be brought to laboratory, where the samples will be analyzed for target element, TDS, TSS, and AN. So firstly, turn on peristaltic pump and collect water for a few minutes. Transfer sample to sample container, and when sample container is full, turn off pump, remove, and quickly fasten the cap of the sample container. Next slide. So done for collecting sample. I will explain about the procedure to transfer sample to sample container. Points that need to be followed are: make sure surrounding area is clean to reduce potential contamination. Use clean pair of gloves. Hold baler or peristaltic pump closely to the mouth of container without contact to avoid air bubbles. Fill container until positive meniscus where it reach at the top of sample container. Fasten the cap, invert, and tap with fingers to check air bubbles. If present, discard and refill new samples. And then preserve samples that need to bring to laboratory. Note that there will be 36 samples where there are 6 replication for each parameter so that we can gain representable data. So for samples laboratory, samples need to be preserved to prevent any changes. Samples will be secured with clear tape to avoid spillage and kept chill with temperature of 2 to 4 degrees Celsius. Next slide. And for disposal, excess groundwater will be properly poured down the sampling gradient, where higher ground elevation is recommended. Next slide. Lastly, error should be avoided to get close to perfect result. This can be done by referring to the guidance provided by US EPA before conducting sample. Use proper equipment, and lastly, as high peristaltic pump pressure may cause interference where salts and other solids may be drawn, so it is recommended to use low pumping pressure. Moving on to the next sampling. Next slide. The objective of this sampling are to determine the water quality parameter of the surface, water and determine the impacts of fertilizer used in the paddy field at Sekinchan on surface water. The water quality parameter includes DO, BOD, COD, AN, and pH. Next slide. So in this slide, it shows the rationale including all these parameters where DO to assess the effect of dissolved oxygen through aquatic life, BOD to determine the effect of the fertilizer on the amount of biochemical oxygen demand, COD to see the amount of chemical oxygen demand that undergoing chemical processes, AN determine the amount of ammonia on the surface water, pH to see the effect of the ingredients in the fertilizer on the surface water. Next slide, please. So, alright, moving on to the procedure in surface water sampling. Firstly, sample collection must be located. Choose a sampling spot of at least 15 to 20 centimeter deep if possible, where you can reach it from a solid spot on the bank of the river or from a large rock. Before that, slide before. Okay, it is advisable to approach the location of the sampling from downstream if necessary. So next slide. Next, the water temperature is then taken. The thermometer will be placed in the water at the point of sampling. Avoid disrupting the bottom of the sample site. It is advised to enable the thermometer to settle, and when the temperature is seen to be static, then it is recorded. Using digital thermometer will be easier as it balances faster and are easier to determine whether the temperature of the water is reached. Next slide. Moving on to the next procedure is label bottle correctly. Sample bottle should be brown, 250 ml, and made from HDPE or LDPE plastic, unless otherwise specified. Then, black permanent marker is used to indicate the location of the sample, which includes geographic area name, date and year, time of the day, and the first and the last name of the sampler. Next slide. To avoid contaminating bottle, put gloves and clean the cap bottle to prevent all possible contaminants. This is because, hold on, please. Okay. To avoid contaminating bottle, put on gloves and clean the cap bottle to prevent all possible contaminants. This is because fingers may carry chemicals like nitrates, insect repellent, sunscreen, and tobacco smoke, which may interfere with the samples. It is crucial to make sure that none of the samplers touch sample bottle with no gloves. Next slide. To prepare the sampling bottle, the inside of the bottle, lid, and the rim are rinsed with deionized water. Next, discarded samples should be discarded downstream or offstream and farther from the sampling site. Lastly, place the cap back on 1/4 turn after the final rinse. Next slide. So before we collect the samples, we need to rinse the bottle with the sample water first for at least 3 times and then continue to collect the samples. In order to collect the samples, keep the cap with your index finger on the bottle, then immerse the bottle about 10 centimeter deep. Don't forget to record the depth of the sample that was obtained. Lastly, remove the cap sufficiently to let the water fill the bottle. Next slide. Next. So the position of the sampling must be taken note, where it must be left and right side of the river where the bottle will be pointing upstream. Next slide. Lastly, seal and store bottle for transport. The sample must be placed in a cool place and do not expose sample bottles to the sun. Next slide. Then complete miscellaneous post-sampling task. Okay, next. Next. Okay, there are many things that need to be considered to obtain accurate results. For example, large non-homogeneous mix. Okay. There are many things that need to be considered to obtain accurate result. For example, large non-homogeneous piece of detritus should not be included. Moreover, avoid touching or disturbing the bottom of a water body when taking sample. Next, ensure the sampling equipment is clean and maintained. And lastly, ensure all field measurement instruments are fully calibrated before starting. There are many things. Next slide. There are many things in ensuring good result to be obtained. For example, sample is taken consistently from the same location may not accurate and presentable. Therefore, we use 3 different locations for the surface sample. Okay, sorry. A bottle that is to be used for transport or storage of the sample should be rinsed 3 times with portion of the sample before being filled. And lastly, sample for pH determination must be separated. The same sample must not be used for any other determination of potassium chloride diffusing from the pH probe. So Atira will be continuing with air sampling.
I am Siti Atira Binti Alias and I will explain about air sampling. Firstly, for introduction, ambient air is referred to outdoor air or of atmospheric air in its everyday state. Here is the table of new major ambient air quality standard. The value of pollutants obtained from this sampling will be compared to the value in this table. Next, active air sampling will be performed. For air sampling, as it will work using a mechanical device like pump the air of contaminant mixture through or into the sampling device to capture airborne pollutants. Next, table shows the types of particulate matter obtained in this sampling and their units. Next, Sekinchan, Rice Bowl of Selangor have rice factories there. Therefore, ambient air monitoring needs to be done to make sure that the presence of rice factory did not affect the API index. The first sampling location was near the rice mill and the second one is at the nearby village. The sampling was conducted from time to time as ambient air pollutants at the sampling location do not come from only surrounding condition but also by activities carried there. For example, trace gas from rice strawberry air at nearby village as wind blows from the paddy field to nearby villages. These pollutants are very harmful. As an illustration, next. From the table shown, we can conclude that PM10 and PM2.5 obtained at the rice mill can cause lung cancer, respiratory disease, and heart failure to people nearby. Next, I will explain about the procedure. The analytical parameter was chosen by looking at the new Malaysian ambient air quality standard and the analytical method by using a high volume air sampler. This instrument was chosen as it can collect a wide range of air. The high volume air sampler, this is the schematic diagram, was set up at rice mill and nearby the targeted village. When the high volume air sampler was turned on, air will pass completely through the sampler and through the filter system as the fan in the instrument start to rotate and works. For TSP, it will be trapped on the filter. Meanwhile, for particles overweight than 10 micrometer are trapped on grease plate. Smaller particles will pass through the instrument onto the free weight filter. The sampling filter containing the pollutants will be removed and transported to lab to be weighed. Their concentration will be determined when dividing the mass by the volume of air samples. Then the result of both locations will differentiate with the value in New Malaysia Ambient Air Quality Standard that had been shown before. Next. The concentration of trace gas was also measured. As for that, trace gas will be collected by using an adsorption method. This method can be used to measure the specific surface area and pore-size distribution of materials. Gas will be collected using activated carbon at the targeted affected village, and the gas sample will be brought to lab. The concentration of the gas sample will be measured using GCMS, mass spectrometry, to identify compounds by the mass of the analyte molecule. Sampling material and packing material, like seal, were being disposed in a correct manner after the sampling was done. Next, to obtain reliable result, preservation steps for ambient air samples, for high volume air sampler, and GCMS must be taken. For example, the sample which are two or more sampler used at given location must be placed at least 2 meters apart so that one sampler will not affect the result of adjacent sampler. Precaution step before, during, and after taking the air samples were also needed to be taken. For example, before the sampling, make sure that the equipment has been prepared properly. Next, an error may occur when conducting the sampling. First, when handling the adsorption method, polluted air should be relatively low temperature. Besides, the distance from sampler to obstacle, it must be twice the height and the obstacle protrudes above the sampler to avoid any obstacle. It is important to pre-weight the filter paper as the weight of particulate matter trapped needs to be calculated. Next, I will explain about emission sampling. Next. Despite the breathtaking view, emission sampling needs to be done in Sekinchan paddy field. The special boundaries are at the rice mill, where the emission were recorded at 40 meter, 80 meter, and 120 meter upwind and downwind from there. The sampling were done from 7 am to 7 pm daily, from 7 November to 8 November, as it was harvesting season that time. Parameters for the sampling are methane, nitrous oxide, including toxic gases such as VOCs, ash particles, and dust particles. Next, the table shows the analysis of this sampling. From here, it can be known the pollutant were measured in which unit. For example, ash particles were recorded in micrometer. This sampling was done in order to determine the amount of pollutant emitted resulting from the activities here, and the samples were collected from areas around paddy fields and rice milling factories. Rice straw is presently disposed of mostly open burning. This results in release of various pollutants affecting the environment. Straw burning also causes pollution. From this table, we can see that the parameter and its rationale based on the location. For example, methane which can be obtained at paddy field as a result of straw addition. Next, for procedure, the information is obtained from literature surveys and workers of the rice mill beforehand. This is done to encourage the collection of suitable analytical method which involve gas chromatography equipped with FID and ECD. Spirometer that will be homogenized with a trowel in a dedicated 1 gallon disposable pail was also used. Each and every analyzer and equipment installed SCEM system should be in compliance with international standards like US EPA. The CEM will continuously operated at all times, collecting and recording valid data. FID can be used to measure hydrocarbon concentration. This is the schematic diagram of FID. When they burn the paddy straw, it will produce, when they burn the paddy straw here, it will produce hydrogen. This method provides high sensitivity and operates a wide range of concentration. ECD, this is the schematic diagram of ECD. It can be used to detect atoms and molecules in a gas. The ECD detects ion by reading the charge in voltage value that maintaining a steady ion getter at the collector. We use for nitrous oxide analysis in this sampling. Next, spirometer Next, spirometer is an apparatus for measuring the volume of air inspired and expired by lungs. It measures ventilation and the movement of air, so the amount of particles that are exposed to the workers can be identified. Next, gases that collected using Tesla bags was tested immediately. Air particles were collected by using tape. Samples were put in a sealed bag. The samples then were taken to UiTM laboratory, plus the sampling waste like used Tesla bag was disposed of in a correct manner after the sampling was done. Uh, Next, few areas were identified during the sampling. The mini volume sampler did not calibrated before use and not in the right position. Also, contamination occurs when the sample is collected. Time taken to do sampling is not precisely 12 hours. The sample do not remain in the sampler in the same duration. So, to prevent this error happen, so that accurate result can be obtained, this can be done by first calibrating the mini volume sampler before use and ensure that it is in vertical position. Next, inlet must be covered to avoid contamination. Ensure that the sampling must be done at least 12 hours and sample must be remain in the sampler for the same duration. To set reliable result, next, precaution step before, during and after the samples were taken. For example, before the sampling, ensure that the mini volume sampler works well. Packaging and shipping were also done in a correct manner. For example, during pre-sampling, the exterior of the sampling glass jar were being wiped first to avoid contaminants. That's all for me. Next to another presenter.
Okay. Now I will be move on for the sediment sampling. Next. For the brief introduction, the major source of heavy metal contamination in paddy field can come from the frequent use of fertilizer and herbicides. Thus, sediment sampling needed to recognize the many sedimentary components, including the amount of metal from pesticides and nitrogen fertilizer. Next. For the objective and the rationale, it is to determine the amount of heavy metals for lead and arsenic in the sediments. The analysis are the pH, total organic carbons, particle size, bulk density, and particulates organic matter. Next. This is a 6 different point of location using systematic grid pattern. SL1 until SL4 is at the subsurface paddy sediments, but each at different depth to check the possible contaminants. For the SL5, it is near the reservoir outlet to monitor the quantity contaminants of each parameter in the sediment containing irrigation water. And for the SL6, which is below the reservoir outlet, it is to monitor the sediment in irrigation water flow out and discharge from the surface reservoir. Next, I will move on for the procedure from the pre-treatment until the disposing. Using the tube or hand auger, the sample taken from areas of slow-moving and no-flow water beneath the paddy plants at a minimum depth of 3 cm and put it into stainless steel or plastic bowl. And then to produce a composite representative of the sample, the sediment is thoroughly homogenized. Volatile organic analysis sample must be directly transferred from the sample collection unit or pool prior to mixing from different areas. It is to reduce contaminant degradation during homogenization due to volatilization. For the containers, we use broad mouth glass containers with Teflon lined caps with cooling to 4 Celsius, since chemical preservation of solids is not recommended. Next, decontaminate all sampling instruments and then wrap them in aluminum foil. For disposing, disposable sampling devices for sediment are practically impractical. Next, we will move on for the error and what is the solution. For the error in the sediment, it can occur from the packaging, includes the existence of a plasticizer in plastic containers, and sample tested for metal components can be treated as a contaminant. Oven drying can turn sealed clay sediments into bricks. Thus, packaging error can be minimized by avoiding contamination. Containers and implements should be carefully chosen. for sediment handling in the field and laboratory. Next, during pre-treatment sample, many standard methods specify the temperature to 100 degrees Celsius, and contamination from dust or the heating elements is a concern, and oven can be flushed continuously with filtered air. Next, to obtain a reliable results, sample must be obtained upstream from the generators or from some other equipment competent of leaking combustion fumes or oils into the sample. Most downstream samples should be obtained first to prevent contamination due to disruption and suspended solids of sediment due to sampling activities, to prevent the chances of contamination between locations. For the precaution, all sampling devices used need to be decontaminated according to EPA Region 9 recommended procedures. It is to avoid any cross-contamination between devices and the sample. Okay, next I will move on for the microbiology sampling. For the brief introduction, Sekinchan paddy field use urea fertilizer to increase the nutrient uptake of paddy plant roots, and certain pesticides stimulate the growth of microbes. But too much use of fertilizer can lead to creating too high of a salt concentration, and this can hurt the beneficial soil microorganism. Those microbiology sampling is needed to know what type of microbe and fungal presence there, and to calculate the microbial growth if there are overuse of fertilizer and heavy metal used at Sekinchan. Next, I will move on for the objective and the sampling rationale. It is to investigate the effect of using fertilizer and herbicides to the soil pH and its impact to the microbial growth in the soil at Sekinchan paddy field. The microbes that are found there is bacteria, nematode, fungi, and actinomycete. The tree location is used randomly since distribution of pesticides and fertilizer is different. Uppermost of topsoil is used with 0 to 30 cm depth since this type of microorganisms live at the roots of the paddy plants. Next, I will move on for the procedure and the pre-treatment. First, the paddy plant is unrooted with the soil forming every plot taken the topsoil 0 to 30 cm depth, put in a clean bag and were labeled. The sample must be transported to laboratory in 24 hours maximum. After that, make dilution of the soil sample including 12 replicate sample, and then 0.1 milliliter of the lowest dilution transferred to culture media agar plate followed by the higher concentration. Repeat the steps with different agar plates for different types of microbes. Neutron agar Na is used for bacteria, and potato dextrose agar PDA is used for fungi to calculate DCFU from subculture the sample. Nematode and actinomycete use nematode growth agar and actinomycete isolation agar. Seal and incubates at room temperature and observe after 24, 48 and 72 hours. Preservation of bacteria can be used using laboratory freezers. The analysis that will be observed were colony plate count and colony growth rate, distribution and number of microbes per gram of soil. For disposing, all the materials including the agar plate will be collected and the material will be put in a stainless steel container and into a vertical portable autoclave. The badium then will be drained with hot and with running water, and then the glassware need to be washed. And for the accuracy of error and the solution, during dilution, the calculation mistakes in the dilution procedure can happen. For the better solution, preparation of the media must be done correctly. Factors that can disrupt the multiplication of the microbial count can be avoided by calibrating the apparatus that will be used to prepare the PDA. And for the precaution, next slide. Disinfect the instrument by using liquid chemicals for surface decontamination. Second, cleaning is required before sterilization or disinfection in removing materials such as soil and organic material. And then, avoid the sample from being exposed to the air before being sterilized to avoid cross-contamination. And lastly, place the petri dish upside down to prevent condensed water from dripping onto the agar and causing colonies to run together. So that's all for microbiology. I will pass on to Dia.
Okay, I will now present about biology sampling of fauna. The introduction of this biology sampling is that it investigates the effects of the urine concentration towards T. granosa, also known as blood cockles or kerang. This sampling uses a total of 6 sampling points to collect the T. granosa samples and study its relationship with thiuron, which is a common herbicide used in paddy fields. The objective, next slide please. Okay, the objective of this fauna sampling is to determine the mortality rate of T. granosa before, during, and after the irrigation water that contains thiuron was released from the paddy field. The objective is to study about the declination in population, mortality, and production of T. granosa samples. This is because there have been previous articles that studied on this issue. Next slide. This is the sampling rationale of fauna sampling. The mortality was observed by recording the number of alive and dead individuals to determine the mortality rate. Mortality rate and population density formula can be seen below. It must be noted that the number of dead individuals is represented by empty shells and those that did not close its shell when being provoked or stimulated. The next slide shows the sampling location for fauna samples. Now let's take a look at the location. As you can see in this map, there is a total of 6 main sampling points, which are P1, P2, P3, and P4. S5 and S6. T. granosa are collected from points S5 and S6, which are the cockle cultivation plots in the coastal area. This coastal area is described as the littoral zone, so it has a higher population of T. granosa compared to the other sampling points. Next slide shows the procedure of fauna sampling. The first step, which is sample collection, requires T. granosa samples to be taken in 3 different sampling events, which are before, during, and after irrigation water was released from the paddy field. They were collected from cultivation plots of S5 and S6, which was shown in the previous slide. Then, a handheld dredge was used to collect the T. granosa samples in a hauling method. The handheld dredge, which resembles a basket, was used because they also have a 1 mm mesh, so it is easier to collect them. Then, hauling was performed several times horizontally to cover the area. After the samples are collected, they are kept in a container and transported to the laboratory. At the laboratory, they will be placed in an aerated tank for 24 hours with water characteristics such as salinity with 30 ppt, temperature of 27 degrees Celsius, and pH of 7.5. Next slide. Now, the preservations and packaging for fauna sampling throughout the study can be seen in this process. So before the sampling takes place, it was made sure that the samples were taken from only the same cultivation plots instead of other sampling points. The instruments and devices used are also calibrated and cleaned properly so that there will be no contamination. During the sampling, samples are kept in a safe container with suitable temperature for it to survive longer. This can be done by using a multi-parameter water quality to measure the water quality inside the containers is safe for the T. granosa samples. The sample protocol is also retained in each of the sampling series. Then after the sampling, the T. granosa samples were packed in a tight and proper container so the samples do not escape easily, since they are still alive. It is also labeled with proper text such as the date, time, and person in charge of the samples. At the lab, T. granosa is kept in an aerated tank, secured with cushioning materials such as cotton and paper towels to prevent it from leaking or breaking during the transportation process. The aerated tank was also made sure to be made out of a solid, durable, and secure material. The next slide, we can see that there are 3 types of waste produced and must be disposed of. The first type of waste is solid waste, which consists of mostly debris and microorganisms. As a way to dispose this, all materials are placed in a stainless steel autoclave. In a stainless steel container. They are then put into a vertical portable autoclave for 20 minutes. As for the glassware, they are washed with hot water following the SOP of the laboratory. The second type of waste is liquid waste, also known as wastewater. Liquid waste from aerated tank that contains seawater are pumped into a huge bag, so it is easier to be disposed of. The sediments and solid waste are filtered and thrown into a proper bin. The third type of waste is chemical waste, such as diuron, methanol, and remaining chemicals in seawater. They are first stored in plastic bottles, which are labeled with the name of the chemicals and their quantity. Next slide shows the field screening of fauna sampling. Now, since the actual sampling did not produce the expected results, field screening is done to obtain a more accurate and reliable result. The first step is to collect the T. granosa samples from a different sampling point, in this case P1, P2, P3, and P4, which are in the water channels of the paddy field. Then they are taken to the laboratory and are placed in an aerated tank. In this tank, they are exposed to a fixed concentration of diuron for 24 hours. Then, the third step involves preparing 5 beakers of 1 liter containing different concentrations of diuron, which are 0.6, 1.2, 1.8, and 2.4 ppm, including a control group of 0 ppm. Next, 3 individuals of T. granosa are placed in each of these beakers for a duration of 72 hours. Every day, they are exposed to fresh diuron twice per day to maintain the concentration, since diuron has a tendency to evaporate. The last step of field screening is to calculate the mortality rate using the same formula shown in the sampling rationale. Then, next slide. Can we go back to the previous slide? The last step is also, it was made sure that the experiment is conducted in 3 replicates with a control tank containing methanol and seawater. Next slide. The last part of fauna sampling is to observe the error and solution. Now, the first error is external contamination. So, as a way to solve this, the T. granosa samples are kept in a proper container and temperature during transport process from the sampling site to the lab. This is to make sure it will survive longer. The second error is uncontrolled sampling site and procedure. Therefore, as a solution, the sampling protocol is minimized. The sampling protocol is maintained to minimize bias, such as using the same dredging device and mode, velocity of boat, and hauling time. Cultivation plots are also made sure to be specific, since it is more difficult to locate an area at the coast compared to an area at the paddy field. The last sampling is flora sampling of paddy samples. Now let's take a look at the brief introduction. Flora sampling uses paddy plants as their medium in order to analyze whether heavy metals are present due to paraquat pesticides. Next, the objective of this flora sampling is to determine and compare the concentration of paraquat pesticides which produces heavy metals such as zinc and copper. The second objective is to determine the accumulation of pesticide in paddy plants using acid digestion method, which is then analyzed using AAS. The next slide. Now, this is the sampling rationale of flora sampling. 3 random plots, which is 1,000 meters square each, have been chosen to identify whether heavy metals are present or otherwise. Only shoot and root parts of the paddy are used to indicate and measure the concentration of heavy metals. This is due to movement of heavy metal from roots to shoots and lastly the grain. It is also because the leaves of the paddy is found to wilt easier and faster compared to the other parts of the paddy samples. This flora sampling utilizes atomic absorption spectroscopy AAS for the 6 analytical parameters or methods, which can be seen in the column below. First is extraction, moisturization, acid digestion, filtration, dilution, presence of heavy metals, and concentration of heavy metals. Next slide. Now, the location, the sampling location of flora samples. Okay, for the sampling location of flora samples, it is being conducted at the left corner, in the middle, and at the right corner of paddy field. The reason why these locations is chosen is because the heavy metals might appear from the pesticides used by the farmers or from external factors such as emission from vehicles by the farmers. Other than that, there is also a factory near the paddy field which can release gases to the air until they accumulate in the root of the paddy. It must be noted that each of these plots contain 3 sampling sites for the sampling event. The sampling location was also chosen randomly plots. The procedure for flora sampling. First of all, we can see that paddy samples were collected using a device called a small core sampler. The paddy samples are then cleaned manually and before being put into a polyethylene bags to get rid of the small debris, small rocks, pebbles, and stones or muds. When they arrive at the laboratory, they are washed with different types of water, mainly tap water. The paddy plants are then cut and separated into root and shoots, and they are now known as subsamples. These subsamples are then dried in the oven at a temperature of 60 degrees Celsius for 24 hours. When the subsamples are dried down, they are weighed and grinded into fine texture and kept in a petri dish before continuing with the next method. Now, the next method uses wet acid digestion method. So, for wet acid digestion method, only 1 gram of subsamples was digested using nitric acid and chloric acid with a ratio of 5:1 until it becomes transparent. The digest were then filtered and diluted with 30 ml of distilled water. Then, the filtrates were analyzed using AAS instrument, and the result of concentrations were compared whether they are at acceptable levels of WHO and Food Regulation 1985. The next slide is sampling, preservations, and packaging of flora. Now, before the sampling, the paddy plant samples were cleaned manually with a dry brush. They are then air-dried in the shade. For the packaging of flora samples, paddy samples were kept in used and clean polyethylene bags to avoid any contamination. They were also properly labeled and wrapped with bubble wraps to prevent it from being crushed easily during transportation. The polyethylene bags were also kept in the freezer incubator that was filled with wet ice to preserve it for a longer time. As for the shipping, chain of custody COC forms were placed in a plastic bag. The paddy plant sample were also packed separately. And then a freezer incubator was sealed using a transparent sellotape. In the next slide, the disposal of flora sampling is that any residue of the paddy are properly discarded into the general waste bin in the laboratory since they do not contain any toxic or chemicals. For the error and solution of flora sampling, the first error is random error. So as a solution, 1 sample is randomly taken in each plot. Then the 2 replicate samples are taken for each parameter from sampling points. For an example, to undergo acid digestion method, a total of 3 samples will be taken, whereby 1 sample is for the actual analysis, while the other 2 samples are used for replication test. The next error is inconsistency in samples. To solve this, paddy samples were ensured all of them are similar in terms of physiological age and size so that the results are more reliable and reproducible. That is all for the tight environmental sampling. Farah will read the next slides to wrap up this presentation.
So for the last part, I'll be going to explain a little bit about field health and safety procedure. One of the most critical elements for our experience in the field work is preparing and training before we arrive. So firstly, we need to make medical preparation, then assemble safety gear which include flashlight, allergy treatment, medication that commonly take such as paracetamol, sunscreen and hat, walkie-talkie, first aid kit, and also personal protective equipment for fieldwork activities such as safety shoes, gloves, and masks. Next, while working, leaders in each group need to chat with their teammates regularly and inform the teammates of any changes to time schedule. Next, phone number of person in charge like office number or field location or university contact should be given to each teammate to contact if any emergencies occur. And lastly, student must work in pairs in all time. Next slide. During working, there will be potential hazard that can cause accident. Due to that, we need to know potential hazard that may happen so that we can prevent it from happening. Some of the major potential hazard is like slippery rocks, as it may cause the sampler to fall into the water, so it is advised to wear appropriate footwear and make sure the shoe soles are not worn out. Next is build up toxic chemicals in water may cause sampler to get sick or diseases. Poisonous plant and animals, participants may get stung by animals or may cause skin irritation, so we should wear appropriate PPE, which include wearing gloves in handling the sample. Working nearby roadside that may cause accident, mechanical failure, for example, no battery in phone may cause problem if someone get lost. Dehydration and heat exhaustion may cause illness, sunburn may cause irritation, and flat tires which can cause car accident. Lastly, it is obligated to bring first aid kit, PPE, allergy treatment, and communication equipment to the sampling site to maintain safety for all participants. And with that, I'm pretty sure that we all have covers all important parts in the sampling plan. Thank you.
Okay, Farah, I think probably it's time for us to move forward to the Q&A session. So can you hear me clearly?
Yeah.
Okay, sure. Okay, now I will, before I give you my question, first of all, before I forget, I would like to comment the performance of the presenters. So I would say after this, every one of us will, or every one of you, we're going to receive a common marks. And I'm happy to say the performance of presentation is above average. However, there are a few points that I would need to remind you during the presentation. Okay, number one, some of you are presented very well. You did your presentation. It's a true presentation. And there are a few also that ended up just slide reading. I think you know who lah. If you read the slide, then I think it's very apparent. So these are the things that I can see clearly. Some of you did present with very minimal words in your slide. So you just read the points in your words and you elaborate using your own sentences, not by reading. That is very important because after this you will be scrutinized lah. After this, when you present, you're not supposed to read the content in the slide, just reading. You cannot. Okay, that's number one. Number two, because of the slide reading, we already know that the student might have limited understanding of the content that they are presenting. So that is why I also that also corroborates, it matches with what I expected, because I saw a few mistakes in the slide which I thought that is quite different than what I usually hear. There are a few mistakes. Usually, like people who would like to present, usually the content is not that does not have too much error. And then the final point that I want to point out is that when you use the language, the tenses that to present, you have to be careful on what occasion they're presenting. So now that you're presenting a sampling plan, so logically you haven't done any sampling work yet. Correct? Correct or not? At this
Yes.
point, when you're presenting the plan, you're not expected to have done anything just yet. So what is the tense that you should use?
Present tense.
Future tense would be more accurate.
Okay.
So the wording will be like this lah. The samples will be collected and sieved before analysis is commenced. So do you hear the word will be, and then the verb will be in past tense. So these are the tenses that you should use. Now, after this, you can look back at your presentation slide and see how many of you uses past tense. So I don't need to check the content lah, because during presentation I already hear more than 50% of you uses past tense. Why? Because the sources that you get from internet, they are all papers or reports that are reporting things that they have done. So when you're using the sources, you also be very careful. So that is a major thing that I've seen lah. The rest of it, you come in well prepared. So before you make this presentation, Farah and Eza has arranged to meet with me to get the tracks right. Before you come and present the points, which I think is quite satisfying. Like in 1 hour, you presented most of the major things that I wanted to hear. So that is why I would say overall, your presentation, just presentation only, not the content. I haven't touched the content yet. On the presentation part, you come in well prepared, so it's above average lah. Exact marks, I would need to subject a few Q&A. Yeah, as you have seen the grading rubrics before, your performance right now is sitting between 7 and 8. So whether or not you can go to 9 depends on how well you answer question after this. Okay. So I think I will go into a few general questions lah, because you have been like presenting 1 hour. So I think I'll just give you a few that I managed to remember. Okay, my first question would be, since you already wanted to detect the potential contaminants from agriculture, and the first one is pesticide, you have mentioned a few that includes diuron and also paraquat. I think this question anybody can answer lah, but if you can, I want to know what is diuron for and what is paraquat for. What is the usage of these two pesticides?
Doctor, can I answer the question?
Yeah.
Usually, from what I know, it is used to control the grasses. It is a herbicide used before and after the plantation takes place.
Okay, now I think perhaps diuron. Wait, which one is that? Is it paraquat or diuron?
Diuron, doctor, because for fauna sampling we mostly focus on diuron concentration.
Okay, so diuron is a herbicide lah. Am I right? Can I say that diuron a herbicide?
Yes. Yes.
What about paraquat?
Paraquat is also one of the herbicide, which primarily used to control wheat and grass.
Okay. Now, at this point, I would say I would not going to dispute your choice of choosing diuron and paraquat, because maybe these two herbicides will not be used at paddy field, but around paddy field. So with that, you will need to clarify. You will need to clarify where the paraquat and diuron will be first discovered, which is around the area of paddy field. Because what? Because if you look at paddy field, they are usually inundated with water. Grass or weed is not expected to survive at all. So technically speaking, I don't see the need to use paraquat or diuron in the middle of paddy field. You will not see in the middle of paddy field, maybe around it. Yes, I admit, can. Now, there is another pesticide that I think should have been given more attention is the use of pesticide like insecticide, I would say insecticide that address the problem of uh, snails. Okay, so anybody from here who is staying at Utara, like Kedah and Perlis, near paddy field, knows that those area they have one very bad pest lah, and that is siput gondang emas. They have been using a lot of powerful pesticide to address siput gondang emas, or in English we call golden apple snail. So if you want to add after this, I think diuron can be included, but the major problem if there is the pesticide used on addressing this golden apple snail. Now, I don't think you need to change too much. It's just the background, the introduction part. You add just one more only, okay? One more insecticide on that one. And that makes the story more understanding. The real problem is actually there, okay? The pesticide. Not to mention also the fertilizers that you have mentioned, so good. I would say these will be the two major problems in Sekinchan. If there are these two problems, and the air pollution. The air pollution coming from the milling factory is also one, but it's a smaller one, and the affected people will be the workers inside the milling factory lah. So in your introduction, you have to clearly state what are the risks and how big the risk is to the community, because it's not logical to say that those dust from milling factory will going to affect the entire population. No, it is not. It's just the occupational hazard, yeah, inside the factory itself. Whereas the other one, you say the dust coming, the ash coming from the burning of rice straw, that could be one. It happens only a certain time. So that is one thing that I will need to highlight, not just on air pollution, the sampling for air sample. The other one part that I want to highlight is the water sampling and also soil sampling. Now, I think during the second presenter who have presented the soil sampling, he mentioned that there is a cycle in the plantation of paddy. Uh, Wet season, dry season. The times where the paddy field is filled with water. Of course, you cannot go in and take soil sample. You can only take soil sample during the dry season, just before musim menuai or immediately after musim menuai, where all the rice paddy is gone. Then only you take the soil sample in the middle of the paddy field. So I think the one thing that is missing in your sampling plan is the timing, because I think in your sampling plan it's unique is because you have to do your sampling according to the planting season, because those who want to collect water sample can only do it during wet season but not dry season. Okay, however, those that are going to take air sample and soil sample have to do it during dry season. So this is the uniqueness of your sampling plan. You have to say exactly what time, when, because I think this season is quite fixed. Right? It's not subjected to too much of changes. It's quite stable. Every month of this coming, wait, if not mistaken, April. April is the musim menuai, if not mistaken. So you have to state, the timing, which month that you want to go sampling. And like I said, lah, water sample, those who with water sampling, air sampling, and soil sampling, your timing are all different. It cannot be at the same time. Okay. Okay. So I move on to the next part lah. That I think you probably need to check, double check is the what? The aquatic organism sampling. Am I right? The one that sampled blood cockle. What was the sampling again?
Yes, blood cockles, Tigranosa.
Okay. Are you sure it's T granosa and not A granosa?
Since we use this article journal, so we just refer to that. And the article mostly uses Tigranosa.
Okay. Because I did the same research before, and I also used blood cockle. I used the shell, and if I remember correctly, the name is Anadara granosa, so it's A granosa. So you better double check. Okay. So you.
But doctor, we use this journal article and the title is Impact of Diuretic Contamination on Blood Cockles, Tachylarca granosa, which is why we are confident to mention it as T granosa instead of A granosa. So is it still okay, or
You double check first. You double check because, like I said, this is just one paper, and paper also is written by human. Human can be wrong. So there's one more thing that I forgot to tell you, because you're not yet in FYP. Once you're in FYP, your supervisor will inform you that not all paper are correct. You have to be careful. Some papers are written wrongly. I'm not sure if this is one, but you have to double check what is the scientific name for blood cockle shell. I did my research before, so I remember it's A granosa. So maybe you want to double check. Okay.
all right, noted, doctor. Thank you.
Okay, so that's one, the name of blood cockle shell. Number two, when you say you want to analyze the migration of pesticide, that is a good idea. But my question is, in the sampling location, you want to collect blood cockle shell along the waterways. So either the river or the water channels. I think I remember correctly that most of this blood cockle shell is located at pantai, seaside, not waterways. Are you sure waterways have blood cockle shell?
Because since the water channel is only used to collect for the field screening, and in the okay, for this presentation and this whole research, we only uses this one journal article, so we mainly rely on the information contained in the journal, which is why we are confident that in the water channel there is Tigranosa too, besides the coastal area. Sorry, doctor, if you're kind of wrong.
I think you may have a point that it's just there are things that you have to weigh in terms of risk lah. While it is known that blood cockle shell majority sits at seaside because that is their breeding ground, I cannot say that the waterways will not have blood cockle. They might be there. The only thing that might be risky here is that might not be as much as you think. So these are the things that you might want to weigh in when you do this sampling plan. So what if the blood cockle cannot be found in the sampling location? Are there any other organisms that you also can collect and analyze? The answer is yes, can. Like your, what you say, I would say mentarang also can be used, not just kerang. So you need to have plan B also. Plan A, blood cockle, can, because why? Many people eat it. But the other one more thing that a lot of people eat also is mentarang, can. So you also can pick that as plan B as if cockle cannot be found, then mentarang is what you need to use. And I think mentarang also is a better candidate, given that mentarang can grow much more bigger. And why? Do you understand why mentarang, when they grow bigger, is better than cockle that is smaller? Anybody can answer?
Sorry, doctor, can you repeat the question? I did
Okay.
not understand that well.
Okay, the question is, I propose to use a bigger mentarang compared to a smaller cockle, blood cockle. Do you understand why bigger mentarang is better in terms of sampling to detect this pesticide? Why? Why bigger is better?
So there will be more concentration of pollutant.
Yeah, you're right. Any organisms that can grow bigger in size should have higher concentration of the pesticide because they grow older, they grow bigger, they accumulate more. So this is true for any other organisms lah, yeah. Now there is another thing that you also need to consider is if there are higher trophic organism in the food chain. So mentarang, above mentarang, what eats mentarang? Maybe there are some birds or maybe there are some predators of mentarang. If you can get that, that is also good because predator of these bottom feeders, they will accumulate even more pollutants. So easy job lah. You just get one of the sample. For sure you can get high concentration and yeah, there is that. But it all depends. Yeah, depends on the sample. So since you already determined cockle, I think cockle is also is a good idea. But there are some pros and cons. So during sampling plan, you also need to consider a few more species. Then only you can have like a backup plan. If plan A doesn't work, then plan B. Because when you go into the field, you never know whether you get cockle or not. So by the time you reach there, you have plan B in hand. So you just go for plan B lah straight away. Takkan you nak sampai sana ada masalah, then only you figure how to solve the problem. Kan? You cannot. You have to figure the problem first and then prepare the solution siap-siap. If the problem is indeed there, then okay lah, you got the solution already. So I think I don't want to like drag you too long lah. Now, just now I asked you a few questions, and it seems like you have answered about half of it. So I think your marks right now is sitting at 8. Whether or not you can go for 9, let's see lah. The other circumstances. I would quite safe to say lah. Your presentation mark right now is sitting at 8 out of 10. Okay, I think 8 out of 10 is very good already, right? Kan?
Yes, doctor.
Yes.
Okay, so I'll give everybody 8 in this case. Now the next part, which is your assignment. You need to submit your assignment, just one copy of assignment. So Farah and Eza arranged to make all the necessary correction, and then I have mentioned to you also a few suggestions right here. You just add in. I believe there's not much you need to correct lah, given the completeness of your assignment already. You just need to correct according to my comments just now, and then you submit a soft copy of the assignment to me, and then that's it for this course. So submit to me lah the corrected version as soon as possible and get ready for your final exam lah. So everybody knows the exact date kan for your final exam.
Yes.
Can somebody tell me the exact date? Exactly
4th
when?
February.
4th of February, that means how many days from now? About 3 weeks lah kan? Is it?
Yeah.
About 3 to 4 weeks.
3 to 4
Yeah,
weeks.
3 to 4 weeks. So the first subject that you're going to, I assume that most of you also take soil science subject. So the first subject for final exam is environmental sampling. The next one would be soil science and sampling. Okay, now I think I'll inform you that this Thursday got test, right? For soil science, am I right? Or is it next
Yes.
week? This Thursday, right?
Yes.
This Thursday.
Okay, now for this Thursday, I think you're also aware that this test will be objective, all objective questions, but as usual, there are some time restriction, and if not mistaken, 45 questions, right? Is it 45?
Yeah, 45
Yes, Doctor,
question
45
for 45
questions.
minutes.
Okay, so these 45 questions will need to be answered and submitted within 45 minutes or 60 minutes.
60 minutes, Doctor, because I think that 1 minute is only for reading the question. Nak fikir tu lama sikit.
True.
Oh, okay. So okay lah. One hour. I think that fits it lah. Now, I cannot stop you from cheating. So if you want to cheat, you can cheat, but I would refrain you from doing it lah. Try your best to answer the best you can possibly can with your own ability. So those will be your tests. Now, in your final exam, there will still be objective question with one essay, or I would say one essay lah, because I cannot mark too many of your script. So the soil science, first thing that you need to address is the test, and then the next final exam will be in your environmental sampling. So in the environmental sampling, it's quite hard for me to give you question. So in this case, it will certainly be online final exam. It will not have objective questions lah. That's quite sure.
Jap. The quiz will be on 21st of January, not 14th.
Oh, so next week lah kan?
Yeah.
Okay, so this Thursday no class, no lecture, so you can have a lot of time to prepare for next week. Maybe for this week you try and finish up your assignment lah kot. Okay, all of you finish your assignment, submit to me as soon as possible. Then next week, the entire week, you just focus on your test lah. Okay. All right. Okay, so I think I haven't finished talking about the format of your final exam for environmental sampling. I would say that will be short, structured question. It is not objective. It is very short, structured question where you need to answer right away. So that is what I can tell you right now lah. All right. So any more question that you want to know about the final exam? I think test you already know lah. Final exam.
Doctor, I have a question. For our final for both EVT 533 and 536, right? Will the papers have the same format as the previous partial questions?
Okay, now I would say it will not have the same format because I am planning to make it smaller. You have 2 hours previously at perfect condition. You physically answer on the marking script. This time, they only allow you 2 hours also. So for sure, I don't think people were going to have enough time to answer the same number of questions as before. Because I need to consider also some of you might be trapped at kampong with your flooding and also the internet connection. So I need to figure out how I would like to format this. But one thing that I can assure you is I will try to make this much easier. I would try to make it less question lah. But there is a drawback. You see, if there are less questions, that means the weightage for each of these questions will be bigger. So if you got wrong answer a lot more than it should be, that means you lose a lot of marks. So I need to warn you first. One thing for sure is there will be lesser question than last year, but the format will be short structured question, just like previous year, but you will see less question. Okay, so that is for environmental sampling. However, for soil science and analysis, I would introduce objective question, multiple choice question, with less question from essay and short structure. Okay, so that is the difference lah that you will see compared to previous final exam. So if you want to ask me whether or not you can prepare using past year question, the answer is yes, and you are encouraged to do so. So is that clear? That's a very good tips, kan?
Yes, doctor, that's really clear.
Yes.
I also have another question. For the submission, is it through Microsoft Team and kena ada percentage of Turnitin juga ke? Like for our lab reports, for our finals
Yeah.
later.
You mean your final exam?
Yeah, for our final exam. How is the submission?
Still thinking about it because I think I would like to use Microsoft Teams. Okay.
But no Turnitin lah, sir.
I think it will connect it to Turnitin. So if anybody copy from website, so I know lah. It won't going to affect your submission. Every one of you are going to submit, and I'll mark later from there. So at Microsoft Teams, we're going to check lah against internet. Where do you get the answer from? Whether from internet you copy from who? I also know. So I will most likely going to use Microsoft Teams the same way that you submit your lab report. But this time, it will give you the exam question, and you're going to answer on the Microsoft Word file. So you're going to type in your answer in Microsoft Word, and then you submit as it is, just like that. Or you may be able to, I think there's only one way, yeah, you submit using that. I'm still figuring out. So at this point, I can only think of Microsoft Teams, but if I can figure out other ways, I'll inform you sooner. The other ways that I can think of is also by using UFuture. UFuture is another option. So either UFuture or the Microsoft Teams. Still finding a way to how to give you this test.
It's okay, doctor, noted.
All right. So any more questions? If not, then I think we can end it today.
Can I ask a question about the insecticide that doctor mentioned earlier for fauna sampling?
Yep.
So doctor wants us to mention about insecticides and about the golden apple snail in our introduction, is it?
Yes, that is correct.
So for the other like sampling rationale, procedure, and the rest of the clauses, is it affected or we just have to change the introduction?
Just the introduction that you need to add about the insecticide for killing the golden apple snail. I think the rest of your sampling would remain the same.
Okay, doctor, that's all. Thank you.
Okay. Any more question?
Doctor, one last question.
Yep.
Do we have to submit the slide?
Yeah. Submit the slide as well.
Okay. That's all.
So you submit the assignment, the PDF, you submit the slide, and also the clip of this video. Let me download this whole video where you presented just now.
Okay.
All right, I think that should be it. So we'll stop right here. But